So Srinivas, thank you again for giving this talk. I'm looking forward to learning some new stuff from you.
Thank you. Thank you for inviting me. You're doing great things already at your Data Science Institute. I see many heavy hitters in your list of speakers in the seminar series.
Thank you. Thank you. Hi, I'm David Bader, and I direct the Institute for Data Science at the New Jersey Institute of Technology. Welcome to this Wednesday's Data Science Seminar, where we're very pleased to have a thought leader in computational biology and data science, Professor Srinivas Aluru. Professor Aluru is Executive Director of the Institute for Data Engineering and Sciences, called IDEAS, and Professor in the School of Computational Science and Engineering at Georgia Institute of Technology. He co-leads the NSF South Big Data Regional Innovation Hub, which nurtures big data partnerships between organizations in the 16 southern states and Washington, D.C., and also the NSF Transdisciplinary Research Institute for advancing data science. Srinivas conducts research in high-performance computing, large-scale data analysis, bioinformatics and systems biology, combinatorial scientific computing, and applied algorithms. He's received many awards, including the NSF Career Award, an IBM Faculty Award, the Swarnajayanti Fellowship from the Government of India, the John V. Atanasoff Discovery Award from Iowa State University, and the Outstanding Senior Faculty Research Award, the Dean's Award for Faculty Excellence, and the Outstanding Research Program Development Award from Georgia Tech. He's a fellow of the AAAS, the IEEE, and SIAM, and a recipient of the IEEE Computer Society's Golden Core and Meritorious Service Awards. So we're very pleased to have Srinivas today. During his talk, you can ask your questions in the chat, and we'll get to those questions at the end of the lecture. So if you're following on Zoom, welcome. And if you'd like to subscribe to our YouTube channel, please go ahead and do so to be alerted of our future events every Wednesday. So thank you again, Srinivas, and let me bring up your slides.
Thank you, David. Thank you for the opportunity to speak at your Data Science Institute. My talk is focused on big data challenges in the area of computational biology, in particular genomics and systems biology. And I understand that the audience who are listening to the talk have a broad variety of backgrounds. So my talk will be geared to audience who may or may not be familiar with this field at all. I'll be happy to answer any questions at the end of the talk, and also if any of the attendees have further questions or interest in reading up more on one topic or the other that I will end up mentioning in today's talk, please feel free to contact me, and we can take this further. So here is an example root cause of where big data arises in the area of computational biology. In particular, you're looking at genomics. All the genomic studies rely on a basic fundamental capability to sequence DNA. Since the 1970s, up until 2005, the technology used for DNA sequencing was called Sanger sequencing, where you do a biological experiment and you can read a fragment of DNA of size about 700 to 1,000 base pairs, somewhere in that range. Now, if you want to read multiple DNA fragments, you have to replicate that experiment. And the high-end machine during those times was the ABI 3700, which had 96 modules where you could carry out 96 of these experiments, each reading about 800 base pairs of DNA. You get a throughput of somewhere around 75 to 80,000 bases. And the cost was about $1 per kilo base. And that was the rate at which DNA was, we could sequence DNA. Now, you could sequence DNA using a variety of high throughput sequencers, which are called next generation sequencing systems, or NGS for short. An example of such a machine is the Illumina NovaSeq machine. It can read 20 billion DNA fragments of size about 150 base pairs in pairs of 150 base pairs each, for a total throughput of about 3 terabases per experiment at a low cost of about $1 per 100 million bases. So when you compare this with the ABA 3700, the throughput of sequencing went up by a factor of about roughly 40 million, and the cost went down by a factor of about 100,000. As a result, what required an international team of coordination for sequencing something like a human genome can now be done by almost any investigator for a few $100s by using the Illumina NovaSeq. As a consequence, huge data sets, which were once a hallmark of international projects, have now moved to individual investigators with small budget. So anybody who has $10,000, $20,000 can do a large genomic study and generate anywhere from hundreds of gigabases to terabases of DNA sequencing data. As a result, everyone needs to have ability to analyze this big data. So how bad can this get? Here is a prediction for the year 2025. As we are live streaming on YouTube, you can see the expected storage needed for YouTube in the year 2025 in terms of exabytes per year. And right next to that, you can see the expectation for the area of genomics, which is somewhere in the range of 21, 22 exabytes per year. So much larger than other technical feeds like astronomy and Twitter and YouTube and etc. So the question is, what do we do with all this DNA sequencing data, and why do we sequence DNA? So first, I want to introduce you to a rich variety of problems that actually arise from this single instrumentation of high throughput or next generation sequencing, or NGS. Typically, to use NGS, what you do is that you have a DNA sample. This could be from a single organism, could be any source, virus, microbes, plants, human, other animals, etc. Or it could be a collection of genomes, like a collection of microbes. So you have a DNA sample consisting of a bunch of very long DNA molecules that you would like to know about, like genomes, for example. The human genome is about 3 billion base pairs in length. But you can only sample very short fragments or reasonably long fragments, but nothing of the scale of genomes, right? So the human genome is 3 billion base pairs in length. Short read next generation sequencers give you 150 base pairs per read. Long read DNA sequencers give you 10, 20, 30,000 base pairs per read. The longest reads that we could ever sequence is in the few mega base pair range, like 2.3 mega base pairs or so. Still, many orders of magnitude smaller than the original genomes. So what you do is that you take a collection of original genomes, you fragment them into smaller pieces. So you randomly fragment them, so you lose the position information. You put them through a high throughput sequencer and you get raw signals like this. The first step in the big data analytics is to take these raw signals and convert them into DNA sequences using a procedure called base calling, augmented with error correction. So we don't get perfect sequencing. So we can interpret these signals, and there could be different possible bases in the same position. So you try your best to do base calling and error correction. And the result is a set of DNA sequences. You can think of them as strings in the alphabet ACGT. Each string corresponds to a read that you read, which is a fragment of DNA, a short fragment from the original DNA sample. You don't know the starting position, and there would still be errors remaining in these sequences. So that's what these samples look like. Now, what we do with these samples next depends upon the application where this is being deployed. Let's say you want to sequence a human genome. So you want to sequence the genome of a specific individual, want to see how this individual differs from other humans and see if that has any implication. For example, many diseases have their basis in genetics, and by sequencing a human genome, we might be able to tell if that person is susceptible to a particular type of diseases. So to do this, given that we know that any two human genomes are almost identical, up to 99.9% or so, We don't need to sequence a human genome from scratch. You take an already sequenced genome, and then you take the reads that you sequenced from another individual, you map them, and computer science-wise, you can think of them as taking a short string and trying to find where that short string would be approximately located in the extremely long string, which is the genome, subject to some natural variations and some sequencing errors. So you have to accommodate for some errors. So you can think of this like a string containment problem subject to some errors. And you can use the reference genome that you know as a guide. You can map all the reads from another individual. Then you can stitch together the mapped regions and come up with the new genome. So this is called read mapping or genome mapping, and tends to be an easier problem than the problem where you have never sequenced any individual from a species. So for example, you encounter a new species of life, a new animal or a new bird or something, and then you want to know its genome. So you fragment the DNA of that species. You've never seen it. So all you have is the reads that you have sequenced to come up with the original genome. So you look for overlaps between these reads. If you sequence a very large number, then you're bound to have sequenced fragments that overlap each other. So you detect these overlaps and you put them together. And this process is called genome assembly, which is a very compute intensive problem because you need to put together hundreds of millions to a few billion DNA fragments to get enough coverage across the genome. And sometimes in scenarios such as where you sequence a bunch of microbial genomes, you get them as a community. There is no way to separate them into individual genomes in the lab. So you collectively fragment their DNA and then you run them through the sequencer. As a result, you are getting snippets of DNA from many organisms. You have no idea how many species were there in the sample. You have no idea what the relative abundance of each species is. But from the sample, you have to try to figure out everything. What species were there? What was the frequency? What are their genomes and so forth? So this is a much harder problem of metagenomic clustering and assembly, one level higher than genome assembly. Another thing that you can do with next generation sequencers is what is called transcriptome assembly. Here, you don't sample the genome, but the genome contains actors called genes, and these genes get translated into RNA, and the RNA gets translated into protein. These are the gene products. And the proteins and RNA interact with each other and then make all the life processes happen. So if you want to understand any biological process, you have to measure the level of expression of these individual genes. So what you do is that you take their mRNA transcripts and then you run them through the sequencer. So you get fragments of them, and then you do clustering of those, and you come up with what are called digital gene expression counts. This would essentially tell you the number of times you have seen a gene expressed. So by taking these expression levels under different conditions, you can figure out which gene products are influencing the expression of other genes or other gene products and how they work together. And how they work together is depicted by networks. What I'm showing you is an example of a gene network, but there are also other networks like protein-protein interaction networks and so forth. So you can derive these networks if you have the gene expression counts under various experimental scenarios. And this forms the foundation of systems biology. Once you have a large number of genomes, we have to figure out how to store them. So there are compression algorithms for these strings, which are basically, genomes are strings with an alphabet of size 4. So you can perhaps design specialized compression algorithms for these. And you also want to index the genomes so that you can do pattern matching, and you can do analytics on them. You can search, you can find, you can collectively analyze them and so forth. So this leads to a very broad and rich set of problems coming out of just next generation sequencing or our ability to sequence DNA. Now, there are other types of high throughput instrumentation used in life sciences. For example, to measure the peptides that are part of a protein, to measure metabolites, or to measure gene expressions through other means like microarrays and so on. The common feature to all of them is that they all end up generating very large scale data sets that must be analyzed. Even within NGS Bioinformatics itself, apart from the richness of the problems that I have already shown, there are other reasons why you have a large set of problems. For example, there isn't a single sequencer. There are different types of sequencers. At one end, you have highly accurate, meaning less than 1% error rate, and very high throughput, meaning in terabases. Very short read sequencers, like you only read 150 base pairs or so. And on the other end, you have long read sequencers, which have generate variable length DNA sequences. The average length may be in the tens of kilobase pair range. The longest ones are Oxford nanopore. Sometimes the reads could be a few megabases. So you have shortest being about 500 base pairs, longest being 1 million base pairs, average length around 10 to 30,000 base pairs. The error rates of these sequencers are high. They're long, but they're like 10% or more than 10% errors. But if you go long range, you can still make sense out of it, even though there are more errors, and so forth. So there are a variety of sequencers because of the length characteristics, differences in length characteristics, error rates, and all kinds of things, and the types of errors they make. Illumina typically makes substitution errors. PacBio makes random insertions and deletions. Oxford nanopore has its own signature types of errors. So you need different algorithms to tackle data that comes out of each sequencer. So that's another source of heterogeneity here. And then the grand challenges, even though the applications may be mapping, assembly, and metagenomic clustering, and digital gene expression analysis, and so forth, the context in which they arise also leads to a rich set of problems. For example, in the cases of viruses like coronavirus, these viruses are fast mutating, so we want to understand who infected whom. The picture that you're looking at the right is a graph depicting the status of a virus in a single individual, where every node is a strain of the virus. They mutate very fast, even within an individual, within weeks. And the two virus strains are connected by an edge if one of them resulted from the other through a single base pair mutation. And the size of these circles indicate the prevalence of that strain. And by drawing large graphs like this, you can tell who is infected from whom and so forth. In the microbial case, microbes are everywhere. They're there in the soil, they're there in the oceans, they're in various organs of the human being. And understanding them really is essential. And so you could engineer them for human needs. And for example, if there is a bioterrorism incident, you would have to sequence the sample there, a community of microbes, and quickly try to figure out what was modified in a dangerous way, and so forth, right? So those are the grand challenge problems on the microbial side. We also want to have knowledge discovery on a broad scale. For example, we would like to sequence the genomes of all the species that we see on this planet. We would like to sequence all the genes of those genomes, try to understand how life functions for every species. In agriculture, we may be interested in engineering plant genomes for optimal yield and natural pest resistance without pesticides and so forth. In precision medicine, we may want to sequence the genome of every individual human being, try to find disease signatures. So for example, we know that women with breast cancer typically have mutation in certain genes called BRCA1 and BRCA2 and so forth. So understanding these disease signatures will help us try to sequence every individual and try to see what diseases they would be susceptible and so forth. So between these different dimensions of variety of sequencers, variety of problem scenarios, and a variety of types of species, you have a very, very rich class of problems with literally thousands of problems that people can work on. And these are all important problems. So as you can imagine, there is an army of bioinformatics researchers, probably thousands in the United States and tens of thousands if you go worldwide, who are working on all of these problems, over the last 15 years or so since the next generation sequencing came about. So what I'm going to do in this talk is basically talk about a couple of projects in my lab. We have undertaken at least a dozen projects over the last 15 years. But the time I have would help me to describe a couple of projects at some length. We actually work in many aspects of next generation sequencing led bioinformatics that I've shown you in the picture before. And in particular, I want to emphasize the work that we do using parallel computers, because when the data is very large scale, a natural way to solve the problems would be using high performance parallel computers. So I would go in that direction and describe a couple of projects. The first project that I want to describe is a 5-year, $2 million project jointly funded by NSF and NIH in their first round of big data investments back in 2012 and 2013. And the goal of this project is essentially to say, okay, now we have perhaps 1,000 or so papers, solving various problems in next generation sequencing bioinformatics, looking at various applications and so forth. Now we are hit with very large scale data. So it would be nice if there is a way to execute all of these bioinformatics tools in parallel. But most of them, an overwhelming majority of them, I would say maybe 95% or so, are essentially sequential software. They're not written with parallelism in mind, because there are a lot of algorithms people and people who write serial software working in this field. So one way to parallelize would be to look at every piece of software individually, and then every sequential algorithm individually, and try to come up with parallel algorithms, and then try to develop a parallel version for every algorithm, every software. So clearly that is insanely time consuming, and there's no way to do this for 1,000 different pieces of work. So the question we asked is that, can we do something quickly, not only for us who work in the high performance computing community, but also for others who may not be very familiar with high performance computing, but is there a way to quickly get them up to speed where they could develop parallel software in the bioinformatics area? So the approach we took is something that's sort of like inspired by BLAS in the case of numerical linear algebra. So for numerical linear algebra, the basis is matrices. So if there are vector, vector, matrix, vector, and matrix, matrix operations parallelized, like scalar pack, for example, then people can build numerical codes very easily in parallel by relegating most of the work to these libraries. So the question we asked is that, can we do something similar in the bioinformatics area? So the area is large, so we really focused only on next generation sequencing area. That sub area, we're only dealing with sequences, which are strings. So we identified string and graph data structures. So these are indexing structures or data structures that are frequently used in solving a variety of bioinformatics problems. And these underpin most computational biology software in the genomics area. So we took these things, they are suffix arrays, suffix trees, LCP arrays, Burrows-Wheeler transforms, FM indices in terms of string data structures. There are overlap graphs, De Bruijn graphs, and so forth on the graph side. And we came up with parallel algorithms for constructing all of these structures and querying these structures. The idea is that if somebody wants to write bioinformatics software, they can essentially use these foundational data structures, their effective parallelization without knowing into any of the details underneath. And then they can add the rest of it. So we built these index structures, and one could also build some core algorithms on top of it, and then leave it there, and the application components can rely on the core algorithms and so forth. So we could do a multi-layered software framework where the higher levels will give you ease of programming, and you can forget more details, and the lower levels will give you higher performance. And even at the lowest level, you can completely relegate the work of data structures and so forth to our libraries. Now, it turns out that the string algorithms in particular are notoriously difficult to parallelize. The main reason is that sequentially, many of these problems can be solved in linear time, which means that when you try to solve them for very large scale data, the computational complexity wise, the work is linear. The data movement wise, the data movement is also linear, so it's as much. So there is not really much of a gap between the computational work and the amount of data that you need to move. So the data movement constants, which tend to be higher, no matter how high the bandwidth is, the computing is obviously much, much faster. So really you're exposed to that, and they become data-driven problems. They're not compute-driven problems because the computation is only linear. As opposed to matrices, right, where the matrix size is quadratic, the matrix algorithms may be cubic, like solving systems of linear equations and so forth. So there is a gap which makes it a little bit easier, and here there is no gap. And so they all become data-bound problems. And before our work, by and large, there were PRAM algorithms for many of these string algorithms. It was very, very hard to solve them in distributed memory. There are shared memory algorithms that work on multiple cores of a node, but it was very difficult to do these things in a distributed memory setting. So we actually were able to do that, and we created a library. And I won't go into details of all of the algorithms, but I'll give you like a couple of details. So a lot of bioinformatics software uses what are called k-mer indices. A k-mer indices are essentially, you take the DNA reads, or you can even take the whole genomes, and you break them into strings of length exactly equal to k, where k is a constant specified by the user. And k is typically a small number. It can range anywhere from 10 to 50 to at most 100. It's not bigger than that. So if you have a genome, you look at every substring of length k. Or if you have a read, you take every substring of length k, starting from every position of that read. So they're overlapping, they're not disjoint. So any substring you find of length k, you take it. So what you do is that you index the set of k-mers that you see in the collection of strings that you're given. And you can also carry a lot of auxiliary information about these k-mers. So for example, you could say, hey, where did I see this k-mer? Which read did it come from? What is the position in the read where I saw it? How frequently did I find this k-mer in my input data set? And what is the guarantee that the k-mer is read correctly? So there's a quality score output by the sequencer for every base that tells you the probability with which that base is correct and so forth, right? So there is a bunch of auxiliary information that you could, and the indexing of k-mers is commonly used in almost every application area of bioinformatics. And k-mers are very simple. The reason is that if you're looking at length k strings, where k is a small number, you only have 4 to the power k variety, right? Because there are 4 bases, and you have k bases in a substring of length k. So you have 4 to the k possibilities. So it's going to be a dense array, and then you catalog, each k-mer, where you saw it, how frequently you saw it, and so forth. And that information is very valuable. So what we did was we built the first distributed memory k-mer indexing library. It's fully customizable, so the user can specify the k is the k-mer, but the values, auxiliary values could be a bunch of things that I talked about earlier. And then we also developed optimized indices for specific values and so forth. And these indices come with standard operations like insertion, deletion, find a particular k-mer, count, and parallel versions of these. So you're given a bunch of k-mers, you want to insert them en masse or delete them en masse, or find the counts of those things en masse and so forth. The algorithms here is pretty simple. It's actually all it turns out is algorithmic engineering. To construct these indices, what we do is that we take the input data in a FASTQ or a FASTA file format. So these are standard format in which you get biological sequence data. And you partition this essentially, and you do parallel I/O. So each node in a distributed memory machine will read its portion, its fraction of the reads. Essentially, you read input of roughly the same total length. You don't care about the number of fragments, but you read roughly the same total length. So you break them down into all the constituent k-mers, and the k-mer space, the number of k-mers is 4 to the k. So you can easily partition that space across processors. So now you can take a look at each k-mer individually, directly hash it to its corresponding number, and then figure out which processor it belongs. So I'm showing you each processor with a different color here. So these k-mers have to go into P0. The dark brown ones have to go into P1. The teal color ones have to go into P2 and so forth. So once you have it, you can do a one operation to shuffle them, and then you can build the local indices. It's fairly simple, and anybody good with high performance algorithm engineering can do this. So we build these indices, and I'm showing you some examples. So the top graphs show you the scalability of our code as you increase the number of cores. The left figure corresponds to the construction time of the index, and the right side is basically cumulative queries. Of course, in parallel, you're not going to do a single query, you do a bunch of queries together. So you can see that it scales well. Also, the rest of the k-mer counters, when we created our distributed memory k-mer counter, were shared memory. So in the bottom table, I'm showing you comparison with them by indexing k-mers from whole genome reads. These are reads that sample whole genomes of chicken, human, strawberry, etc. On the right side, you see assembled genome. So here we took the full human genome or the full pine genome and took k-mers from that. And you can see that Kmerind, the library that we created in distributed memory, actually significantly outperforms even shared memory software within a single node in a shared memory setting. On top of that, we can also deploy multiple nodes within distributed memory. And if you want to read more about this work, it's published by my student and now research scientist Tony Pan is the primary author. It's in the ACM BCB conference 4 years ago. Also, there is a IEEE publication in Transactions in Computational Biology and Bioinformatics in 2017. So you could consult these. The string data structures like suffix arrays and suffix trees are actually pretty hard. My group has worked for several years to come up with very satisfactory distributed memory algorithms for these. So let me introduce to you what these data structures are. So suppose that you have a single DNA fragment. I'm showing this as S equals CA, TTA, TTA, GGA. We append $1 at the end of the sequence, not because we like money, we do like money, but the reason we append $1 is to make sure that when you have a collection of sequences, or even when you have a long sequence, we want to make sure that if I pick any two suffixes, they differ at some point. We want to avoid the situation where a smaller suffix is a complete prefix of another suffix. So by putting a unique character $, at the end, by the time we hit the end, every suffix would be different. And that's important to create this string data structure. Now a suffix tree, which is the figure that I'm showing here, is basically you take every suffix of this sequence. A suffix is a substring that starts at some arbitrary position, and starting from there, you read all the way till the end of the sequence. So a suffix can be uniquely specified by a number. So if I number the positions in the string, starting from 0, 1, 2, 3, etc., suffix starting at position 0 would be the entire string. Suffix starting from position 1 would be you skip the first character, but the rest of the string, and so forth. So you can see that in this tree, every path from root to a leaf corresponds to a suffix. And you have suffixes described according to their numbers in the leaves. And if you look at the leaves, actually, if you take each suffix to be a word in a dictionary and sort them in dictionary order, that's called suffix sorting. And that gives you left to right order of leaves in the suffix tree, which is called the suffix array. And there is an auxiliary data structure called the longest common prefix array, where you take every consecutive pair of suffixes and you find the length of their longest common prefix, which also happens to be depth of their lowest common ancestor in the tree. By equating them, you compute them very quickly. And these data structures are really, really critical. They allow you to solve a variety of bioinformatics problems extremely fast. They're also very complicated algorithms, so I don't have the time to go into them in great detail. But let me give you a little bit of a flavor. So suppose you want to construct a suffix array. Then all you have to do is you have to describe each suffix with a single number, which gives the starting position of the suffix in the string. And you have to sort them in dictionary order. And sequentially, you can do this in linear time. Amazingly, this is a sorting problem of a set of strings, which have a particular structure because the strings are all the suffixes of a string. So you can do this better than sorting numbers. That would take n log n time, but here you can do it in linear time. So the input, the total length of all the suffixes is actually quadratic, but you can describe them using a linear set of numbers, and you have to sort them. So here is a quick idea about how to sort them. So what you do is that you sort all these suffixes by one character, then you double it. You go from 1 to 2, 2 to 4, 4 to 8, etc. So in log n iterations, you can converge. The basic idea is that here is an example where I'm showing all the suffixes sorted by 2 characters. I'm showing you the actual suffixes for illustrative purposes, but what we store are just the numbers that I'm showing you at the top. So this is a partial sorting where everything is sorted by first 2 characters. Now I want to extend sorting by 2 characters to sorting by 4 characters. So the way I do that is to recognize that if I take 2 suffixes that are sorted by 2 characters, and if I suppress the first 2 characters, the remaining are also suffixes, and those suffixes are sorted already by their first 2 characters. So by taking the current array positions as primary key, and where the remaining strings go as the secondary key. And by doing a radix sort, you can extend it from 2 to 4. So you do a linear time sorting, and then you can converge in log n iterations. And you can do this using parallel radix sort, and then you can come up with some shortcuts. You don't have to start with the base case of 1. You can start with the base case of maybe 10 or something, and then double very frequently. And you also don't have to do log n iterations. In practice, after you do a couple of iterations, you would have differentiated all the suffixes, because as soon as two suffixes start to differ, you know their final position. So the repeats are what creates a problem here, and the repeats will not be that long to span the entire genome. So these theoretical and practical optimizations converge very quickly. So I want to go back to the previous slide and to show you results. For example, for indexing the entire human genome, which is 3 billion base pairs, we could take the human genome, which is the S here, and we used a fairly standard Xeon cluster processors coming maybe like 4 or 5 years ago. This is a 2015 best paper at supercomputing by my PhD student Patrick Flick. So he was able to construct suffix array and LCP array for human genome from the genome in 7.5 seconds using 32 nodes, 16 cores each. And then in another 1.7 seconds, he was able to construct the suffix tree from the LCP and suffix arrays. This tends to be an even more complicated algorithm that I would really hate to go into in a short amount of time. But the basic idea, there are 2 key ideas here that you might like to know. One of them is that once you have the suffix array, you have all the leaves in the suffix tree. And one thing that you can readily see once I tell you is that if you take every pair of consecutive suffixes in any tree, doesn't need to be suffix tree. If you take any tree data structure and you take every consecutive pair of leaves and then ask what is the lowest common ancestor of this consecutive pair of leaves, and you generate a list of all those lowest common ancestors, you would have generated every internal node in the tree in one stretch. You would generate each of them multiple times. For example, if you look at the node V here, it has a bunch of children. And if you take any consecutive pair of children, like say U0 and U1, if you look at the rightmost children in U0 subtree and leftmost child in U1 subtree, those two are consecutive leaves, and their lowest common ancestor would be V. So this way, every internal node at every level would be generated in one swoop by taking lowest common ancestor of every consecutive pair. And in the case of suffix trees, it ends up being the lowest, the length of the longest common prefix, which is the LCP, which we already have. So you could use this neat transformation to compute all the internal nodes in one parallel operation, and also set up all the parent and child pointers by solving a nice problem called all nearest smaller values problem, by finding some attractive properties about these longest common prefix lengths. And you can come up with a very, very satisfactory algorithm. Again, I'll show you times, and in both the suffix array and suffix tree cases, our algorithms either show scalability to a much larger number of cores or work much faster, or both. So this work, along with other work to index and query the different string data structures and also different graph data structures, was part of my PhD student Patrick Flick's dissertation. In fact, just this year, his dissertation was picked for the ACM SIG HPC Doctoral Dissertation Award as the best doctoral dissertation in HPC that was concluded last year. Now, if you're interested in this work, all of the software is available at this GitHub address, github.com/parbliss, which stands for Parallel Bioinformatics Library for short sequences. And you will find a bunch of software tools there, both construction of suffix trees, LCP arrays, suffix arrays, and some of the graphs, k-mer indices, and many other pieces of software. And we have also done some workshops to talk about these libraries in the past, but the GitHub is the right resource if you're interested. And we would like to popularize the use of this library, because if you're a bioinformatics tool developer and if you're interested in writing parallel software, you could relegate much of the work to this library, and you can build your applications very, very, very easily. In the remaining time I have, I want to go over switching gears. I want to talk about an interesting application in systems biology. I already mentioned to you that to understand a biological process, you need to figure out what genes play a role in that biological process. You have no idea. There will be tens of thousands. So if you don't know anything, it would be guesswork. And then you have to do a bunch of experiments. There could be time series experiments, could be taking a bunch of organisms of the same species, try to do it in the absence or presence of the biological condition to see what genes are expressed more and less and how it changes with time. And then you build this network. Now, this network is very useful because you have to understand the function of a gene, you have to catch a gene in action, right? So for example, if I take cells from your brain, from your gut, from your pancreas, from your heart, and so forth, and come up with gene expression analysis, and I see that some genes are differentially expressed in these different organs. If some genes are expressed more in the heart than anywhere else, then you know that these genes have some role in the functioning of the heart muscle. So that's how you do the biological guesswork. So as I mentioned, we would love to know the genomes of all the organisms, but more importantly, also about every gene in every organism. What role does it play? How does it work? So here is a motivation, an example plant, Arabidopsis thaliana. It's a model plant. It's actually the simplest plant organism that you can think of. Its genome is fairly small, 1/8 of a gigabase, 125 megabases, sequenced in 2000, first model organism to be sequenced, has about 22,500 genes, and each gene can create multiple protein products. So you have about 35,000 protein products. So NSF, the National Science Foundation, launched a program called Arabidopsis 2010, right after Arabidopsis was sequenced in 2000. And the goal of this 10-year program is to fund biology investigators so that everything about this genome organism can be figured out. So in particular, you want to find out the function of every gene. So NSF spent about $265 million over 10 years, and there are European agencies which funded additional research. About $500 million have been spent just trying to understand this organism, which is actually just a weed, by the way. It doesn't do anything productive, but it's a simple plant model organism. Now, fast forward to 2020, the status today, even 10 years after the initial program concluded and there was more funding that was directed in the next 10 years, we still have no clue about the function of a quarter of the genes. So the question we want to ask as data scientists is that, is there a way we could help put the money to better use, maybe learn more with less money and so forth? So what my group, in conjunction with our collaborators, conceived of is the following. There are a large number of gene expression measurement expressions, these digital gene expression analysis or micro experiments that were done. And every experiment basically says, okay, here are the conditions, we did an experiment, and here is the expression level of every gene in the organism. And these expressions are submitted to public databases. So we scavenged all these public databases, downloaded tens of thousands of experiments. So you can think of it like a gene expression matrix, where the rows are the genes. So you have 22,500 rows, one for each gene. Then the columns are conditions. So if you have 20,000 experiments, you have 20,000 columns. So you have a huge matrix. And can we construct some kind of a coarse genome scale network? So create the network from the expression matrix that you observe. And you don't expect these networks to be perfect. But the question is, can we use this to derive intelligent hypotheses? So there are obviously, by the time we started working, there are a large number of methods to construct network from gene expression matrices. And as you would expect, simple methods were not very accurate or applicable, but they would work extremely fast. And complicated methods like Bayesian networks were NP hard. They would give you some of the best accuracies out there, but they're NP hard. You can apply them for 20 or 30 genes. Unfortunately, we have 20,000 genes. So on top of it, there is a survey paper that basically shows that the accuracy of these methods is not that great. So you really have to go for highly accurate methods like information theory and Bayesian networks to get a reasonable level of accuracy, particularly for large-scale data sets. So what my group did was we went ahead and tried to develop massively parallel algorithms for Bayesian network structure learning. These are general algorithms. You can use them for anything. Even though we use them for gene expression networks, you could use them in any different application scenario. We targeted something called score-based Bayesian network structure learning. The way this works is the following. Assume that you are given a network N, and I'm showing you a small 5-node network here, A, B, C, D, E. What you do is that you decompose the network and say, for each node, what are the parents? And then you're given a scoring function S that tells you the fitness, how good it is to choose this set of parents for that particular node. And it's an additive function. So you compute a score of the network by summing over all nodes the fitness score of the choice that you made for the parents. And the way you pick the optimal network is you can take all possible networks, score every one of them, and pick a network that maximizes the score. A network here is a directed acyclic graph, so you have a super exponential number of directed acyclic graphs. So you cannot really enumerate all the networks and score them. Excuse me. So what we did is we came up with a massively parallel heuristic algorithm where we say, I'm going to compute pairwise correlations between every pair of genes or nodes. And I'm going to not look at every possible parent for a node, but only limit my attention to what I call a candidate parent set, right? There has to be some good level correlations. Excuse me. So what we do is generate a candidate parent set X for every node X, much smaller size than the entire set of nodes. And if this set size is smaller than a threshold T, we enumerate all possibilities. If it is larger, what we do is that we do this computation for all the smaller ones and then say, hey, if somebody claimed me as a parent, I don't want to claim them as my parent because they would likely be my child. So using this argument, we can reduce the set sizes. If the original set size was large, we can make it smaller. And then in the second round, we can address all of these other sets. And then because we made this decision independently for every node, you could have cycles in the graph, so you have to detect and break the cycles. So what you can do is basically the basic building block of computation would be taking the candidate parent set and finding the optimal parent set from that. So you can map this computation as a subset lattice. So what you're seeing at the bottom of this lattice is the candidate parent set. And any subset of this could be your real optimized parents. So you have to take all subsets of this. And that's where you get the subset lattice, which is also equivalent to a binary hypercube. Then you can compute each node independently in the lattice. But it turns out that if you compute a larger subset from a subset that is contained in it, but one smaller in size, it would be faster. So we compute them in a depth-first search manner in this subset lattice. And when you have a large subset lattice, we define a minimum unit of, a maximum unit of work to be an R-dimensional hypercube. And if your work is bigger than that, we break it into smaller hypercubes. And that way we create parallelism. And then we let each sub-hypercube be computed independently over all the genes, over all the sub-hypercubes coming from all the hypercubes and so forth. So we adjust the granularity of this network. And here I'm showing you experimental results on the Tian-He2 supercomputer. This was done back in 2014 timeframe. This was a supercomputing best paper finalist from the applications category. In the graphs that I'm showing you number of compute nodes on the x-axis and the time to solution on the y-axis. And it's a log-log scale. So a linear line means that it is linear scaling. And each node here has 192 cores split between the Xeon main processor and the Xeon Phi processor. So at 8,192 nodes, you're really looking at 1.5 million cores, multiplying that with 192. And we could generate these large genome scale networks that nobody could construct earlier within minutes by using this supercomputer. So what do you do once you construct these networks? I want to show you a quick example. So let's say a biologist is studying a specific pathway. In this case, my collaborator is studying what is called a carotenoid pathway. So the biologist has some idea about the pathway, which I'm showing to the right. They know some genes that are involved in the pathway. And so we take those genes and identify them on our whole genome network. I'm showing you a part of the network, and the green labeled nodes are the known genes. And then using them, we use a PageRank style algorithm and extract a subnetwork. and ask which nodes are there in this subnetwork that we know nothing about. Those are the yellow ones. So these are the genes for which there is no function. And through association in the network, we deduce that maybe they have some role in the pathway. So how do we prove this biologically? So we generated biological hypothesis using computing, data science. So how do you prove it? So to prove it, because it's a plant, it's easy. We call the seed bank and say, can you send me seeds knocking out this gene, AT1G 56500? Can you also send me some genes, some seeds knocking out the other gene? Then we go ahead and plant it. And here is your visual proof. The first plant is the wild type, growing healthy. And we knocked out one gene here, another gene here, and then you can see that they are showing stinted growth. And the carotenoid pathway has something to do with the growth of the plant, so you have your visual proof. You can go more detailed and do molecular analysis. You grind the leaf tissue of these three. You can do digital gene expression analysis experiment and come up with this type of a graph. You see clusters of 3 bars. So the 3 bars correspond to, the first bar is wild type, and the next 2 bars are the 2 different genes knocked out. On the x-axis, you are looking at genes that are knocked out, genes that are known to play a role in the carotenoid pathway. So these are known genes. So the idea is that if your unknown gene works in the pathway, it would have an effect on the expression of the known genes. So the bars are actually showing the expression level of the known genes. So for example, if you look at the ZDS gene, in the wild type, and if the first gene is knocked out, the expression is relatively stable. But if you knock out the second gene, the expression of ZDS went down to 0. So there's an inverse relation. So you can see that there is a proof here that this gene, the second gene, is playing a role in the carotenoid pathway. Not only that, by figuring out which known genes are affected, which are not affected, you can actually place it in the pathway diagram. These are the pathway diagrams, and you can figure out a way to place. For example, if all these genes are affected and these genes are not affected, then it must be in the left branch and not the right branch. So you can position it as well. Okay, so this is how we could actually use data science to drive, generate biological hypothesis and use that to drive. So these are examples of 2 projects in my group. We have a large number of projects. And of course, there are many, many people who work in this area. So there's a lot of work, interesting work in the NGS bioinformatics area. I hope that I showed you some interesting work. Obviously, this type of work can only be done with a large number of current and former students from my group, as well as a number of collaborators. These collaborators span statistics, data science, machine learning, biology, life sciences, medicine, and so forth. So I just want to acknowledge all of my group members and collaborators and the funding agencies. My work is largely funded by NSF and NIH, and in the case of those libraries that I showed you at the beginning of my talk, those were funded by a grant from Intel as well. So I'll conclude my talk here. And David, if there are questions, I'm happy to answer those.
Thank you, Srinivas, for a great talk. If there are questions, please enter them into the chat. I'll have a question to ask, and then I'll read you some questions from the chat. So I'm very curious, you are a pioneer at the confluence of data science, high performance computing, and biology. How did you get involved with working on biological problems when your background and training comes from the computing area?
Yeah, for me, David, actually, it's a case of a sheer accident. In 1997, when I was a young assistant professor, I was working at New Mexico State University, and I think both you and I were in New Mexico at that time. You were at the University of New Mexico. I didn't know anything about biology, but ACM was holding their first Recomp conference nearby in Santa Fe. So out of curiosity, I went and listened to some of the leading players in the field. And what I found there really attracted me. And I noticed that there is a lot of work sequentially, and I only found like about half a dozen papers to read in terms of high performance computing in this area. So I started with that curiosity. I said, why don't I try to take this field and systematically develop algorithms? And then within a decade or so, the next generation sequencing came along, and everybody wanted to do high performance computing. So that accelerated the work in my group. And so we've been working for 20 years, and I hope that we were able to contribute a substantial body of work in that time frame that's useful for everyone.
So that's great. I see a question in the chat from Dr. Jihua Du. Since every suffix tree algorithm can be systematically replaced with an algorithm that uses a suffix array enhanced with additional information, such as the LCP array, and solves the same problem in the same time complexity, why would we build suffix trees that will need more memory than this suffix array?
That's an excellent question. It's a question from somebody who understands these data structures in depth. We do the same thing. We use these suffix tree diagrams for presenting the work. But in fact, what we actually build is what is called an enhanced suffix array. This is suffix array with auxiliary information to simulate anything that happens on a suffix tree. In fact, the distributed memory version we call a distributed enhanced suffix array, data structure called DESA. That's what we use for querying of these data structures. Sequentially, there is excellent work on this topic and software written by Stefan Kurtz, for example, in particular, gets very widely used. And we have a parallel version of exactly the same thing. Even in designing algorithms, people typically tend to think of the algorithms in terms of suffix tree because it's a natural way to think about it. But when it comes to implementation, we all go back to that enhanced suffix array. So we do that as well.
So Srinivas, thank you for a very exciting talk this last hour. I really enjoyed seeing the work that you're doing and appreciate having this conversation with you. So I hope you do well, and I hope you continue to do this exciting work.
Thank you very much, David. And thank you for those who are directly tuning into the Zoom, as well as listening to it on YouTube. If you have any questions or want to probe further into any aspects that I talked about, my email address is my last name, Aluru, A-L-U-R-U, @cc.charlie.georgiatech.edu. And I'm happy to take further questions or any other inquiries by email. Thank you.
Thank you. And we'll see everyone next week on next Wednesday's Data Science Seminar. Take care.
Thank you, David. And thanks everyone for attending.